This is an overview of an article entitled Genetic Susceptibility Loci for Cardiovascular Disease and Their Impact on Atherosclerotic Plaques, published in the September 2018 issue of Circulation, Genomic and Precision Medicine. Atherosclerosis is a chronic process involving a complex interplay of lipid uptake and vascular remodeling, leading to coronary artery disease, or CAD, myocardial infarction, and stroke. Through large-scale consortia-based genome-wide association studies, numerous genetic loci have been identified for CAD and large artery ischemic stroke, or LAS. Histological and physiological studies of atherosclerotic plaques have identified the key components such as vascular injury, endothelial dysfunction, increased vessel permeability, monocyte adhesion, migration with activation of macrophages within the intima, and smooth vessel proliferation. Smooth muscle cells and macrophages engulf lipids, forming foam cells, and recruit T cells, forming fatty streaks in the arterial systems, which can result in chronic ischemia. Furthermore, certain atherosclerotic lesions, termed vulnerable plaques, are more prone to rupture, destabilization, and occlusion through platelet activation, resulting in acute myocardial infarction and stroke. The genetic contributions to CAD and LAS resulting in acute destabilizing lesions are unclear. In this study, the authors leveraged three independent deep phenotyped biobanks: the Athero-Express Biobank Study, the Stockholm Atherosclerosis Gene Expression Study, and the Biobank of Karolinska Endarterectomy Study, to assess the genetic architecture of histologically analyzed plaque specimens, plaque-derived DNA methylation, and tissue-specific differential gene expression. First, the authors tested the association of 61 established susceptibility loci for CAD and LAS with plaque characteristics in the Athero-Express Biobank Study, including the numbers of macrophages and smooth muscle cells, intraplaque vessel density, intraplaque fat, calcification, and collagen. Second, they tested the association of common variants in a locus on chromosome 7q22 with carotid plaque DNA methylation and tissue-specific gene expression by combining data from the three studies. Further details about the three studies can be found in the online version of the paper. 21 out of 61 established risk variants nominally associated with a plaque characteristic. As seen in this figure, RS1253945 in a locus on chromosome 7q22 was strongly associated with intraplaque fat. This variant had previously been shown to be associated with CAD, and the CAD risk allele was associated with reduced intraplaque fat. This intronic single nucleotide variant is located in the gene encoding conserved oligomeric Golgi complex subunit 5, or COG5. Overall, intraplaque fat was the plaque characteristic that was most commonly associated with CAD risk loci. Further characterization of the same chromosome 7q22 locus showed tissue-specific effects of RS1253995 and other linked variants on carotid plaque expression of the gene encoding HMG box transcription factor 1, or HBB1, a nearby gene in the locus, as well as COG5 expression in atherosclerotic arterial wall, internal mammary artery tissue, and whole blood. The results for HBP1 are shown in this figure. There are also effects on methylation in this locus in carotid plaques. Additionally, the authors accessed data from public datasets and observed an association of the variants with decreased LDL cholesterol and increased HDL cholesterol levels in the blood. Because of the central role of atherosclerosis in CAD and LAS and the polygenic nature of these complex diseases, the authors conducted polygenic score analyses and tested the contribution of modestly associated variants, ascertained through genome-wide association studies, on plaque characteristics. These showed the strongest correlation of a CAD polygenic risk score with intraplaque fat. In contrast, polygenic scores for LAS associated with intraplaque hemorrhage and smooth muscle cell number. In summary, 21 out of 61 established cardiovascular disease risk loci associated with human plaque characteristics. Variants in a chromosome 7q22 locus were significantly associated with fat and carotid plaques. These variants were associated with expression of COG5 and HBP1 in different tissues, as well as methylation in carotid plaques. In conclusion, these data suggest that some cardiovascular disease susceptibility loci might exert their effects by influencing atherosclerotic plaque characteristics.